This is the Professor, CBMD PhD, and today is Tuesday, August 4th, 2026. It is 5:04:41 p.m. Eastern Standard Time, and today's research, ovarian cells. So let's hop to it here. Common sugar may help cancer cells break free and spread. Date August 4th, 2026. Source: the Worcester Institute. Chemotherapy surviving ovarian cancer cells may release fructose to help neighboring tumor cells loosen, escape, and spread. The discovery suggests that surgery diets and cholesterol-lowering drugs could influence cancer progression, though the effects have not yet been confirmed in patients. The researchers found that fructose serves as one of the chemical messages released by these treatment-surviving cells. The discovery reveals a previously unknown way that cancer cells left behind after therapy may contribute to metastasis. Some cancer cells that survive chemotherapy aren't dividing anymore, but they are still biologically active. Instead, they continue to release molecules that send signals to nearby cells. Our study is among the first to show that a nutrient, in this case fructose, can act as one of those signals. Nearly all ovarian cancer patients receive platinum-based chemotherapy, and the initial response is often strong. Even so, the cancer returns in most cases and typically spreads throughout the abdominal cavity. This process, known as metastasis, is responsible for roughly 90% of deaths from the disease. Previous studies have indicated that cancer cells surviving chemotherapy may help drive recurrence by releasing a complicated mixture of signaling molecules. To investigate this possibility, researchers developed an experiment that separated the surviving cells from the substances they released. The researchers and scientific investigators collected molecules produced by chemotherapy-surviving cells and exposed other cancer cells to them. Those released substances alone were enough to significantly increase the cancer cell's ability to spread. As far as we know, this is the first time anyone has shown in a preclinical model, rather than just a dish, that it is the molecules these cells release, not the cells themselves, that drive the cancer spread. The researchers then set out to determine which released substance was causing this effect. The analysis showed that the surviving cancer cells produced fructose and used it as a signal that encouraged neighboring cells to spread. The team also found that high amounts of dietary fructose, comparable to the levels present in sugary drinks, could encourage cancer spread even when chemotherapy had not been given. This result raises the possibility that dietary habits may influence how cancer progresses. That possibility is especially notable because fructose is widely consumed. In some people, high fructose corn syrup accounts for 8 to 20% of daily calorie intake. Unlike many risk factors that patients cannot change, fructose intake can be reduced through dietary choices. Researchers have not yet tested whether lowering fructose consumption improves outcomes in patients. Still, the findings suggest that nutrition may affect cancer progression in ways that were previously overlooked. The team next investigated how fructose makes cancer cells more likely to spread, using several large-scale analytical methods, including a CRISPR screen. They discovered that fructose lowers cholesterol production inside neighboring cancer cells. Cholesterol helps cells remain attached to one another, functioning like a form of biological glue. When cholesterol levels fall, those cellular bonds weaken, allowing cancer cells to separate more easily and move into other areas. This mechanism offers a possible explanation for how a common nutrient can alter the physical behavior of tumor cells and increase their ability to escape. The discovery that reduced cholesterol production may encourage cancer spread also has potential clinical significance. Statins work by lowering cholesterol production. In the study, statins alone weakened the connections between cancer cells and made it easier for them to escape. The researchers are now investigating whether these drugs can interfere with the effects of chemotherapy. However, the team emphasizes that the findings are not a reason for patients to stop taking statins or any other prescribed medication. We haven't tested this effect in patients yet, but it raises questions about combining cholesterol-lowering drugs. with chemotherapy, especially since ovarian cancer is most common in postmenopausal women who are often already on statins. The researchers are also studying whether the same fructose-related pathway could play a role in cancers beyond ovarian cancer. They think other cancers that spread within the torso, pancreatic, colon, liver, could behave similarly. They can't call it universal yet, but they think the effects are not just limited to ovarian cancer. Researchers and scientific investigators have already begun planning follow-up studies to determine whether the results can be reproduced in several other types of cancer. This videography was created for educational purposes only. My code name is Will. I'm Professor Christopher Brunet, leader of research scientist, ufologist, psychic, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover. My passion is world peace. Have a great day. This is a Professor, CBMD, PhD, and today is Tuesday.